Technological development in sample collection using GCMS has enabled the collection of large amounts of environmental data. However, the current data reduction methods are highly labor-intensive and time-consuming, which bottlenecks the information gathering processes. Here, we present an automated approach of cataloging and potentially identifying all analytes in a large chromatographic dataset of ambient aerosols, using a matrix size reduction technique followed by a decision tree. This approach can greatly increase the speed and accuracy of the analysis. The core algorithm of this approach consists of 5 different steps. The first step is applying positive matrix factorization, or PMF, on a small subsection of multiple chromatograms. Here you can see a roughly 10-second window of 4 chromatograms. These subsections will then be combined and PMF will be performed. PMF focuses on the data pattern that is repeated over the entire set of chromatograms and yield the valuable patterns as factors. Once factors are obtained from the first subsection, the same process is done on the next subset of chromatograms with overlapping retention time until the entire range of chromatograms are covered. The next step involves extracting peaks from the factors obtained by PMF. In this example, the time profiles of 3 factors are displayed. The goal of this step is to extract all the potentially important information from each factor with minimum filtering. This allows saving and utilizing potentially important information that are often discarded by current conventional analysis methods. The extraction is done by fitting all the peaks found in each factor, and we found a total of 8 peaks from factor 1 in this example. In the third step, the extracted peaks are evaluated based on criteria provided by the decision tree, such as peak size, shape, and location. Lastly, all the filtered peaks from each subsection are combined and assessed to remove the repeated peaks in the overlapping region of the subsections. In order to assess the accuracy of the method, a sample of a known mixture was analyzed. Multiple alkanes from tetradecane to hexacosane are injected in the samples, and corresponding deuterated alkanes are injected as an internal standard in the blank solution. This figure presents the total signal of a chromatogram with injected alkanes. After the analysis was done, the analytes found mainly from the blank solution are presented here in blue, and the analytes found from the samples are shown in red. The results indicate that all the injected compounds were identified along with additional compounds, which could indicate the presence of contaminants. This figure presents not only the injected analytes but also the ability of this algorithm to successfully separate co-eluted peaks. Here at retention time roughly 350 seconds, tetradecane and deuterated tetradecane are presented here in red and blue respectively. The major difference between those two is the existence of the smaller peak co-eluted with the deuterated tetradecane. A deuterium is replaced by a hydrogen due to its relatively unstable nature, which caused detection of a different molecular structure by GCMS. The mass spectra of C14D30 and C14D29H at both base peak and the ion peak regions show the same fragmentation pattern with masses of C14D29H being shifted by 1. The isotope effect is expected to be observed from deuterated alkanes but not from N-alkanes, and our work has been able to capture this with high accuracy. After validating the accuracy of the software using control samples, analysis of a real-world ambient air sample was performed. Samples were collected near an urban area surrounded by a forest located in the central Amazon. 4 chromatograms are used, and a total of 606 analytes were found within a 1-hour period of analysis. These results show a significant improvement in analysis efficiency when compared to the conventional manual analysis of the same dataset performed by a skilled technician, which yielded approximately 150 analytes in several hours.